thank you for the introduction. I'm Felix, and yeah, let's don't waste time. Yeah, I'm pretty sure you know all of this. So the germicidal effects of UV radiation are known for quite some time. The oldest paper I found was almost 150 years old. But even though it's widely used in research and industry, it's not used in occupied spaces so far, mainly because of UV-induced health effects, especially on the skin and eye. But novel light sources and especially the COVID-19 pandemic kind of changed that, and so this is basically why we are here today. And most of the safety studies with skin and also cornea, as we saw earlier today and yesterday, involve histology. And the downside of histology is that it's invasive and often slow, and also especially with skin, the stratum corneum often gets separated from the rest of the sample. And so in this talk, I'd like to show you our findings on the comparison of a Far-UVC lamp with the UVB source. And for imaging, we were using a technique called dynamic-microscopic optical coherence tomography, which was used on an in vitro skin model. And the advantage of that technique is that it's comparable fast, label-free, and non-invasive. So how does it work? So it's based on a kind of it's a fiber-based Michelson interferometer. So basically, briefly, light from a supercontinuum light source is bandpass filtered and then split into a reference arm and sample arm. In the sample arm, the light is scanned onto the sample using galvo scanners, and the interference signal is then detected by a custom spectrometer. And the downside of microscopic OCT without the dynamic processing is that, especially in relatively small fields of view, the contrast is very low, even though the resolution is high enough to resolve cells and nuclei with an NA of 0.3 due to the homogeneous scattering properties within the field of view. The contrast is quite low. And to overcome this, instead of just recording one single image, we record a time series. And this is shown here in this video. I hope you can see this flickering on in this slide. And this flickering is caused by subcellular motion of structures that we cannot resolve, but they affect the speckle pattern, which is a result of the coherent nature of the light that we were using. And then we perform a pixel-wise frequency analysis and calculate the integral of 3 different frequency bands and assign those to the color channels of an RGB image, meaning so the blue area means there is not much going on, green are low to medium frequencies, and red high frequencies. And now, we can see already a great contrast between the viable cells and the stratum corneum. And due to the shallow focal depth, we have to acquire multiple images by shifting the focal plane or the microscope objective axially and then combine them. And once that is done, we get really nice images, as I said, label-free, showing the whole epidermis and also parts of the dermis, the individual layers of the epidermis, and even individual nuclei of the keratinocytes. But this depends on where kind of the cross-section, or if the cross-section hits the nucleus. Our light sources were the KR222 B1 illuminator. So we measured for Far-UVC. Also throughout this talk, when I refer to UVC without Far-UVC, I mean 222 nanometers, because this was the only wavelength we were using. And the UVB source also contains a little bit of UVA, but the most of the energy is part of the UVB spectrum, and that's a mercury vapor-based lamp. And we characterized our light sources with a spectrometer that was kind of freshly calibrated and designed for UV applications. The exposures. Okay. So for the exposures, we oriented on the old and new threshold limit values for 222 nanometers. So we chose 50 millijoule per centimeter squared because it's like approximately 2 times the old value, the old limit, 250 millijoule because it's approximately 10 times the old value, then 450 millijoule per centimeter squared because that's slightly below the current or proposed or newly proposed threshold value, and also 2 joule because this is substantially above both limits. And so during the exposures, we placed the media of our sample, that in vitro human skin model. So the media is this red-orange liquid with PBS to avoid any like potentially toxic photo products in the media. And also very important for the exposure with UVC, we kept the surface, the certain corneum of the skin model dry, so that there is no absorption of the potential liquid layer on top. And imaging was done as soon as possible after the exposure ended, and then 1 hour, 4 hours, 24 hours, and if the sample survived, even 48 hours after the exposure ended. And the images, so after combining images of multiple depths into one nice image where everything is in focus, and denoising, I kind of manually segmented at least 15 cells on the nuclei in each image. And then the images were converted from the RGB color space into the HSV color space. And then only the hue channel of the HSV image was further used, because then the main advantage is that this kind of boils down the color to just one single value. And this is kind of shown here. This is kind of the mask of this image, but only the hue channel. And then the hue value distribution was is shown using violin plots later. And so we are analyzing the nuclei and cytosol separately. And yeah, okay, then let's start with the controls. So I decided to enlarge the start and endpoint a little bit. So those images, they basically look the same, which is great. The only difference is that the stratum corneum here is a little bit thicker compared to the beginning, which is also a good sign, because that means that the keratinocytes are alive and well. Okay, now UVB. So using 50 millijoule per centimeter squared didn't show any effect, but higher doses or higher exposures are causing an effect even after 1 hour, which is not shown here, but on the next slide. But you can see that the nuclei, that they tend to become more orange even after 4 hours, and especially with after exposure with a 2 joule. And after 24 hours, the cells are basically dead. There was still some signals from nuclei left, but the cytosol is barely even visible, probably because it's already kind of broken apart. And so those are some images from the previous slide, a little bit larger, to see those effects a little bit better. So those kind of change of morphology, and also the shift from the more green color of the nuclei to red is increasing as well as with dose and also with time. So this is kind of clearly a sign of the UV damage. And now the interesting or more interesting part why we are here today. UVC doesn't show any damage or any damages or changes. The only thing which happens again is that the stratum corneum becomes thicker after 48 hours, or even after maybe 24, it's visible, which is again a good sign, because that means that the keratinocytes are healthy. Further analysis, so the hue value distribution in the cytosol didn't change at all in the control. Also, that wasn't the case after exposing the samples with 50 millijoule per centimeter squared, but with 250 millijoule per centimeter squared and upwards. We see changes in the cytosol, especially after 24 hours, and which is, as I said, likely caused by apoptosis, shown here with a tunnel stain. So there are lots of cells tunnel positive after 24 hours after exposure with a higher dose, and nothing there with the control. For the nuclei, the hue values were kind of more confined to this region. Therefore, I zoomed into the hue axis, and again, with the control, there are no changes. The hue value is more well located into the reddish, not red, yellowish green color. And the exposed samples, with the exposed samples, it's different. So UVB, so with 250 millijoule per centimeter squared and above, after 4 hours, the hue value kind of distribution curve is more skewed into the red color or reddish hue values. Again, meaning red means kind of more subcellular motion. And then after 24 hours, when the cells are dying or about to die, there are only kind of the nuclei left, but there's not much cytosol visible. So this could even be like an apoptotic body. I'm not 100% sure. But also most importantly, UVC, here's a little artifact, but with UVC, the hue value distribution stays the same throughout the whole experiment. So there are no, or at least it looks like UVC doesn't induce any changes with our imaging technique in that in vitro model. Yeah, to conclude, with dynamic microscopic OCT, we are able to visualize the UVB-induced photodamage again without any labeling. And in the cytosol, there's a shift from high or medium frequencies to very low frequencies, meaning there is not much subcellular motion going on anymore. And with the nuclei, yeah, there's a slight shift from medium to higher frequencies. And also given the fact that the redshift after the high UVB doses is larger after 4 hours compared to 1 hour, it's kind of a good indicator that this is this is not like the CPDs itself what we are seeing, but the cellular response to the CPDs. Shown again here, a little bit larger, so lots of cells with very red nuclei, and there was no reaction in the Far-UVC exposed samples. Yeah, this doesn't there's no contradiction with anything that was presented previous today or yesterday. But there were also no effects with 2 joule. So either, as David Welch said yesterday in his talk, those threshold limit values are still too conservative, or the stratum corneum. So the thickness of the stratum corneum might be kind of also an important factor, because in those in vitro skin models, there is no shedding. So the stratum corneum here is approximately 50 micrometers thick, so about 3 times as thick as an in vivo human skin. So this is definitely something that when you were working with MATEX samples or other in vitro models in the past, and you will see that it's kind of important to maybe reconsider the findings, because this is, even though it looks really nice, but the thickness of the stratum corneum is, it's not normal. And in the future, because manually segmenting cells is always very tedious and honestly not fun at all, we are using a machine learning approach. So far, I only did like a layer segmentation using a unit, which was very similar to the original unit paper from 2015. And the cell and nuclei segmentation is work in progress. And I mean, since cornea safety is also very important, so in the future we will also image cornea. This is a human ex vivo cornea which wasn't exposed or treated at all, and we can see the stroma, the Bowman membrane. I believe we can even distinguish the basement membrane on top of that, and then the viable cells on top of that are kind of also show like a different morphology depending on where they are located. Thank you very much.
Thank you. An interesting technique. Yes, we have a question up there.
Hey, thanks for the talk. I was just curious, would you also be able to use this technique in vivo, like in human subjects, or what does the machinery look like to use that DM OCT?
It's not a handheld device. It's mounted on an optical table, and also like a mechanical setup is very crucial because so that frequency analysis. So we are basically performing a Fourier transformation for each XY position along the time axis. And you can imagine if there was macroscopic motion, then it's off. And then the images don't have that nice contrast, and they are just blurry and white. So we tried some in vivo imaging so far, and the images were not as great that we'd like to show them here. But we are working on that, and there are also other groups that are already using this technique for in vivo imaging. Yeah.
Excellent, thank you.
Anyone else? Okay, thank you very much.